Good morning, students. Today we will see general physiology, 5 minutes, and we will try to cover some 5 points. Number one is in cell physiology, you have to know about the function of the organelles, and the very important is the cell addition molecules. Okay, cell addition molecules are integrins, cadherin, selectin, connexin. These are the cell addition molecules. And apart from that, intercellular junctions, that is, there are three type of junctions: tight junctions, adherent junctions, gap junctions. And the adherent junctions is of two types: desmosomes and hemidesmosomes. And second point is cell membrane. So cell membrane, it is made up of two components: phospholipids and proteins. Phospholipids is lipid bilayer, and proteins are two types: integral proteins and peripheral proteins. And you have to remember the transport across the cell membrane, that is active transport and passive transport. Active transport is transport of substance against the gradient, and it is of two types: primary active transport, secondary active transport. The primary active transport is against the gradient with utilization of ATP, and secondary active transport is, it is also against the gradient, but here the energy is driven by the primary active transport. So secondary active transport is of two types: co-transport and counter transport. Co-transport is if the substances are transported in the same direction, it is co-transport. If they are transported in opposite direction, it is counter transport. And example for primary active transport is sodium potassium ATPase pump. For co-transport, it is sodium glucose transport. For counter transport, sodium hydrogen exchanger. So examples you have to remember. And coming to passive transport, it is the transport of substances along the gradient, and from substance transport from higher concentration to lower concentration, and it is of two types: osmosis and diffusion. So osmosis is movement of solvent from a region of less concentration of solute to higher concentration of solute through a semi-permeable membrane. And diffusion, it is of two types: simple diffusion and facilitated diffusion. So if the transport occurring along the gradient and it is diffusing through the small pores in the membrane, that happens in lipid soluble substances, that is simple diffusion. Example: oxygen carbon dioxide transport across the alveolar capillary membrane. And facilitated diffusion is here also transport occurring along the gradient but with the help of a carrier. So it is a carrier mediated diffusion. Example: GLUT, GLUT, it is a glucose transporter. And coming to cell membrane, the third point you have to remember is resting membrane potential. It is a potential difference existing across the cell membrane, and it is usually negative. So RMP of a skeletal muscle is minus 90 millivolt, of a nerve is minus 70 millivolt, of a cardiac muscle is minus 80 millivolt, and of a smooth muscle is fluctuating minus 45 to minus 55 millivolt. And regarding the action potential, it is a sequence of change takes place in the resting membrane potential after the application of a threshold stimulus. And the action potential curve have a stimulus artifact, a latent period, then a slow depolarization, rapid depolarization, then rapid repolarization, slow repolarization, and hyperpolarization. And here the depolarization is due to sodium influx and repolarization is due to potassium efflux. And coming to the fluids, so inside the cell you have to remember the intracellular fluid, outside you have extracellular fluid. These two forms the components of total body water, which constitute 60% of your body weight. And extracellular fluid in turn is divided into two: one is interstitial fluid, other one is plasma volume. And regarding the measurement, the intracellular fluid cannot be measured directly. It can be measured from total body water and extracellular fluid. And the interstitial fluid also cannot be measured directly. It can be measured from ECF. Okay, so ECF minus plasma volume. And you have to remember the substances used for measurement of the body fluids, and those substances should not be toxic, easily metabolized and excreted. Mannitol, inulin, sucrose used for ECF. And plasma volume is measured by Evans blue. Deuterium oxide, tritium oxide used for total body water. And then fourth point is homeostasis. Homeostasis is maintenance of a constant internal environment through two feedback mechanisms: positive feedback and negative feedback. In positive feedback, when a parameter is increased, mechanism operate and it will increase further that parameter. Example, clotting mechanism. One clotting factor activating the other clotting factor until the process completes. Other example, parturition. So descent of the head, oxytocin released, contraction of uterus, again descent of the head. This is a positive feedback. And negative feedback is blood pressure regulation. Increase in the blood pressure sends signals to the medullary centers and it decreases the blood pressure. So this positive and negative feedback forms helps in the homeostasis. And fifth point, last point to remember is pH. So pH is the pH of plasma 7.4. Any fluid less than 7.4 is acidic, more than 7.4 is basic. And what is acid is a proton donor, what is a base, it is a proton acceptor. And you should know about some acid base disorders. Thank you. So these are the things you have to remember in general physiology. Thank you.